Hello guys and welcome back to Accenture Academy. So we'll be doing today cell cycle checkpoint. This is the last topic in the cell cycle. So it is highly regulated sequential process which is monitored by number of checkpoints. So the checkpoints control function to ensure the incomplete or damaged chromosomes are not replicated and that critical stages of the cell cycle are completed before the following stages are initiated. So in short, what is the function of these checkpoints? To ensure that there is no damaged DNA which is getting replicated and and those with the errors, the DNA mutation or any kinds of mutations are not passed on to the daughter cells. Now, so for G1, as I mentioned before also, so we have got this start point, right? Or we also say as the restriction point. So this is the first stage where you have got the checkpoint. Okay, so that is known as the G1 checkpoint. So it is going to ensure that the cell size is correct, nutrition is available, growth factors is there, and there is no DNA damage before it enters the cells, enters the DNA replication phase. Okay, so first checkpoint is your G1 checkpoint. Then your DNA synthesis takes place. Then we have got the G2 checkpoint. So in the G2 checkpoint, again, it will check for the cell size and the DNA replication. So it is going to check whether any DNA replication based error has occurred or not. If so, then it is going to arrest the cell at G2 and then going to repair. Okay. Then we have got the spindle assembly checkpoint. So this is in the M phase. So here it is checking whether all the chromosomes are attached to the microtubules or the spindle apparatus or not properly. So these are the various checkpoints at which the cell is, regulating its all the process, whether the cell is ready for the particular step or not. Okay, so there are three total checkpoints. G1 and S checkpoint, G2 and M checkpoint, and metaphase and anaphase. That is the spindle assembly checkpoints. Okay, please remember this. Now coming to the G1S checkpoint. So DNA is replicated in the S phase. The cell cycle arrests in the G1 phase of the DNA if the DNA is damaged. Okay. Now this DNA damage could be because of the gamma rays or any other chemical modification. So in the normal conditions, okay, the level of P53 is low. But if the DNA is damaged, then it increases in its concentration. So if you remember, we have talked about if there is a single stranded DNA damage, then ATR gets activated, right? And if it is a double stranded DNA, the ATM gets activated. Okay, so they all result, if you remember, ATM is responsible in the activation of your P53, right? So it is going to phosphorylate. Now, P53, it stimulates the transcription of P21, MDM and BAX genes. Okay, see P53 एक्टिवेट हुआ तो मतलब ये अभी आपका एक्टिवेट हो रहा है. BAX genes क्या है आपके? Apoptotic genes हैं. Now, so P21 is your CKI. What is CKI? Anyone? Krishita, what is CKI?
Cyclin kinase inhibitor?
Not cyclin kinase, it is checkpoint kinase, which gets activated by ATR, right? If you remember, ATR activates your check 1, ATM is for the check 2, then they all get together, activates your CDKs. Okay, so it is a checkpoint kinase inhibitor. It inhibits CDK activity, where CDK is your cyclin dependent kinase and prevents the replication of damaged DNA and arrests the cell at the G1 stage. ठीक है, so अभी हमने क्या पढ़ा? There is a DNA damage. Okay, now this damage could be because of any kinds of gamma radiations or chemical modifications. This results in increased concentration of P53. This is going to activate or increase the concentration of P21, MDM protein and the BAX gene. Okay, now P21 क्या है? It's a checkpoint kinase inhibitor. So it is going to inhibit the CDK activity. Okay, and because of this, what will happen? The cell is arrested at the G1 stage. What CDK activity we have in the G1 stage? CDK4, 6, correct? In the G1 stage, CDK4, 6 with your cyclin D, right? So this activity is now prevented and the cell gets arrested in the G1 phase. Okay, now, so the MDM protein works as E3 ligase for P53. It increases the concentration. It increases the, sorry, one second. It increases the MDM protein expression, which in turn degrade P53, thus providing a negative loop. तो यहाँ पे क्या हुआ है? P53 ने एक्टिवेट किया MDM और MDM ही इसको डिग्रेड कर रहा है, ठीक है? And that's why we say it's a negative loop. So the proteasomal degradation of P53 through polyubiquitination by ubiquitin ligase MDM results in low transcriptional activity of P53, thereby providing a negative loop. So P53 ने इसको activate किया, ये इसी को ही inhibit कर रहा है, ठीक है? So in case of extensive DNA damage, P53 induces cell death by activating genes like BAX and VAX that are apoptotic signaling. So BAX genes are meant for your giving the apoptosis ka signal. Okay. So if in case the DNA is not repaired, then the cell should undergo apoptosis. Okay, so different protein kinases like ATR, ATM, it detects DNA damage. This we have discussed. And these protein kinase phosphorylate P53, increasing the transcription of P21, resulting in blocking the cell at G1 as checkpoint. Clear? So 3 cases were happening when P53 was getting activated. So first we talked about the CHK1, right? Then MDM, and then about the BAX gene. So this is going to prevent the activity of CDK4 and 6 at your G1. MDM is going to result in the low expression or low concentration of P53, which is the negative feedback loop. BAX genes kya kar rahe hain? If they are getting expressed for longer time, then it is going to give the cell signal of apoptosis. Okay. Now, so G1 checkpoint is controlled by many different stimuli, including DNA damage, TGF-beta, contact inhibition, replicative senescence, growth factor withdrawal. So here contact inhibition means what? If you have got the culture plate and यहाँ पे आपके cell divide हो रहे हैं, okay. Now after some time, what will happen? There won't be any space remaining. Right? So when they come in contact with each other, they understand that now no more space is available. Okay, so then they stop dividing. Coming to the TGF-beta, okay. So TGF-beta is your transforming growth factor beta, which controls the proliferation and cellular differentiation in the cells. So we have talked about TGF-beta pathway, TGF-beta SMAD signaling pathway. Okay, so it act as anti-proliferative factor in normal epithelial cells. So it is secreted. It's a secreted protein which exists in 3 isoforms, beta 1, 2, and 3. Now, TGF-beta, it prevents phosphorylation of RB. Now, guys, this is about the retinoblastoma. Okay, so the TGF-beta is going to prevent the phosphorylation of your RB. Okay, which is scheduled in late G1 and arrest the cells in the G1. TGF-beta activates SMAD protein, which in turn activates your CKI. So abhi kya hua? Again, TGF-beta is going to activate. It is going to activate your CKI. CKI activate hona yani ki no CDK activity. Hence, what will happen? No CDK activity. Hence, the cell is getting arrested at G1 phase. Okay? Additionally, it inhibits transcription of CDC25, which is a phosphatase which activates the cell cycle kinases. So remember, CDC25 क्या करता था आपका? Phosphatase, it removes your threonine and tyrosine 14, 15, right? Activating your cyclins. For this reason, cell cycle kinases, which is bound to cyclin E and D, cannot phosphorylate your your RBP protein, right? So what will happen then? Finally, it is activating your CDK activity. Is it clear? Do you remember how retinoblastoma was working? Shall I move?
Ma'am, can you summarize things?
Okay. So do you remember that RB is your tumor suppressor gene? This much do you remember?
Yes, ma'am.
Yeah. And RB, okay, so retinoblastoma is a tumor suppressor gene. So this is bound to E2F. This is your transcription factor, right? So क्या होता था? When in G1 phase, this is there, and all this your CDK4, 6 bound to cyclin D is getting activated. Okay, this complete complex goes and phosphorylate your RB. Upon phosphorylation, what will happen? It loses the affinity for the E2F. So this will get dissociated. Now your E2F is free. It enters the nucleus and results in the gene expression. Okay. Now what kind of gene expression here we are talking here about the cyclin E. Now why cyclin E is required? So that it enters your S phase, where cyclin E will bind to your CDK2 to form a complex. Okay. And that is your S phase. Now, for this slide, what TGF beta is doing? TGF beta is preventing the phosphorylation of RB. So this phosphorylation is not happening. A2F is still bound to RB. No gene expression, no cyclin. So no S phase. So the cell is still in your G1 phase. Now it is clear?
Yes, ma'am.
Okay. Now coming to the CDC25 also. So in the CDC25 case, it is a phosphatase. If you remember, we have spoken about that there are 3 types of phosphorylation happening by the V1. Okay. Threonine 161, threonine 14 and tyrosine 15, I guess. Yeah, ये threonine 15 है, tyrosine 14 है. ये थोड़ा मुझे याद नहीं रहता. But these two are there, right? So these two are removed by your CDC25. And then the complex gets activated and enters your M phase. Okay. So what they're doing? Even the CDC25 is inactivated. ठीक है? तो कुछ भी आपका कोई भी complex ready नहीं है. It's just saying that everything is stuck at the G1 phase. Okay. Okay, here same thing. So we have got the TGF beta. We have got different types. Type 1 and type 2 is there. So it's a serine threonine kinase domain, right? So what is happening upon binding of the growth factor? So what is the growth factor over here? Transforming growth factor. Okay, it goes and binds to your TGF beta receptor. It gets activated. They have got the autophosphorylation. So once this autophosphorylation happens, that is going to result in your activation of your SMAD protein. Okay, SMAD proteins are getting activated by getting phosphorylated. So if you see here, the SMAD proteins are getting phosphorylated and they form the dimer. Now, this goes inside the nucleus, binds, and there is a gene expression of these P16, 15, 21, 27. So they are nothing but the CDK inhibitors. Okay. Now coming to the DNA damage. So P53 is inactive in normal cell and bound to MDM2 protein. MDM2 is an E3 ligase which degrades the P53. It is activated by UV, oncogenes, and drugs which damages the DNA. So P53, we know that it is important. Again, what is P53? Is it a tumor suppressor gene or it's a oncogene? क्या है P53?
Tumor suppressor gene.
Correct. It's a tumor suppressor gene. Okay. Now, when you say that it is bound to M2 protein, what is M2? It is E3 ligase. E3 ligase क्या है? It's a part of your ubiquitination proteasome, right? So M2 का full form, it's क्या है? It is mouse double minute 2 protein. I don't know. I know it's weird name, but yeah, it's mouse double minute 2 protein. Okay, so just remember that M2 means it is the E3 ligase, which is going to degrade the P53. P53 it gets activated by UV or oncogenes or the drugs, which is going to damage the DNA. Now, the damage of the DNA, it is going to activate your ATR, ATM, which in turn is going to activate your checkpoint kinase 1 and 2, which will phosphorylate P53. So, this will prevent the binding of P53 with the M2. So P53 is bound to MDM2 protein. Okay. Now, upon phosphorylation, what will happen? There is, so they get dissociated from the M2. So once activated, P53 works as transcription factor for P21, which bonds the complex of your G1 S CDK and D CDK by inhibiting their activity. And P53 will also inhibit the angiogenesis. So far, so clear, guys? Clear? See, there is a P53. Okay. It is bound to your MDM2 in normal cases. Okay, this much is clear, right? Now, P53 is getting activated whenever there is a DNA damage. So let's say that there is a DNA damage. It is activating your P53. Okay, a P53 activate कैसे होगा? Because DNA damage gets activated. Sorry, DNA damage is detected by ATM. ATM is going to activate your CHK1 and 2. Now, these are nothing but checkpoint kinases. Kinases means it is going to phosphorylate your P53. Correct? Now, this P53 phosphorylation will prevent its binding from MDM2. इतना clear है? हाँ, अब क्या होगा? MDM2 इसको छोड़ देगा, right? This will leave it. Now this phosphorylated P53 is now present. Okay, it is acting as a transcription factor. This much is clear. When I say transcription factor, it means this is going to enter your nucleus. Correct? And it is going to works as transcription factor for P21, which bonds the complex of the CDK by inhibiting their activity. Okay, so what is happening then? All those apoptosis, cell cycle arrays, all those signals will be, or the gene expression will take place. Clear? Now, P53 also inhibits angiogenesis. So this is with respect to your tumor. Okay? So tumor also DNA damage is there. So there also activation of P53 is there. And it should stop angiogenesis so that it doesn't spread. Okay, here we have got the figure. So DNA damage, UV, DNA damage is happening. It is activating your ATM and ATR. Okay. Now there again the kinases which activates your CHEK1 and CHEK2. Again, it is activating by phosphorylating CDC25. Okay. Now here it is also activating your P53. Correct? P53, when it gets activated, acts as the transcription factor and enter the nucleus. And this also we have discussed, guys. Aurora A and B activating your APC. Remember? Yeah. And then when CDC is there, it is going to activate your CDK1 by removing those extra phosphate group. So when CDK1 and cyclin B, this is nothing but your MPF. maturation promoting factor or mitosis promoting factor at the G2N phase, right? So when they get activated, it will result in the transition from G2 to M. Clear?
Yes, ma'am.
Yeah. So for all this thing, like we have, studied so many pathways, right? So all these are coming together. तो ये थोड़ा सा confusing हो जाएगा, but that's okay if you remember the functions of RB, functions of P53, check 1, check 2, CDK1, 2, कौन सा cyclin कौन से phase में है, then it becomes easy. Okay, so that's why I said it is important. Okay, now the growth factors. So guys, I'll be completing this topic today itself, okay? With that, a cell cycle will get over. Okay, so growth factors, activation of proliferation. Many growth factors are involved in proliferation of the cells. For example, IGF-1 and FGF2. IGF-1 and FGF2, insulin-like growth factor and the fibroblast growth factor 2, they enhance cyclin D and E association. So when you talk about cyclin D, it means what? Cyclin D से हम बाद में cyclin E में जाते हैं. So when we say it like this, it means from G1 to S we are going. ठीक है? So IGF-1 was required for G2M progression. FGF2 also decrease the levels of the CDK inhibitor associated with cyclin E CDK2. So cyclin E CDK2 is present in your S phase. Now, uska inhibitor कौन है आपका P27 or KIP1? This will be removed or decreased by the activity of FGF2. So in short, we are saying that when a cell is present and we have got FGF2 growth factor, it binds and results in the decrease of your P27, which results in increase of your CDK2 cyclin E complex. It means हम S phase में चले गए हैं. ठीक है? Now, PDGF, platelet derived growth factor. It induces the formation of your cyclin D CDK4 complex. Okay? Now, again, it is going to reduce the level of P27, which is an inhibitor of this complex. तो इसका मतलब क्या? I am in G1. P27 रहेगा तो I will remain in the G1. So if I don't have P27 or decrease, then cyclin D CDK4 complex is free, it's active, and I can progress from G1 to S phase. So in short, how many growth factors? I mean, there are many, but FGF2 and your PDGF is helping, okay, in progressing from G1 to S. IGF-1 is helping in G2M progression. Again, RB, so, little repetition will be there just because they are the proteins involved in the prevention. So RB is a tumor suppressor protein expressed throughout the cell cycle, but it is phosphorylated during the G1 to S transition. So if there is a DNA damage, you know that RB is getting activated and arrests the cell at the G1 stage. The unphosphorylated form of RB is anti-proliferative. It inhibits the transcription of genes which is required for cell cycle progression. Okay? So it binds to the E2F family of proteins. It's a transcription factor, right? Now it will enter, it will result in the synthesis of your cyclin E. That is when the cyclin D to E it is going, it means the cell is going from G1 to S phase. Okay. So when it is bound to E2F, the complex acts as the growth suppressor and prevents the progression through the cell cycle. Now, it also attracts HDAC protein to the chromatin because HDAC modifies chromatin to close it through deacetylation. Transcription is repressed. Now, what is happening? Just ulta. Abhi phosphorylation of RB is there. So E2F is free. It is dissociated from the RB. It becomes active. Now this is going to, so when E2F is free, it activates factors like cyclins A and E and push the cell from G1 to S. Correct? So this is done by pushing the cell through the cell cycle by activating cyclin dependent kinases and activating pCNA, which speeds the DNA replication and repair by helping to attach the DNA polymerase. Now, guys, थोड़ा बहुत mole bio भी यहाँ पे आया है. So, if you remember pCNA, it's your, what is pCNA? Shruti?
Clamp loader.
That is?
Clamp loader.
Yes, correct. Clamp loader, right? So that's how it is helping in the speed of the DNA replication. So now again, look at the story here. See, RB was bound to E2F, correct? Now, once RB gets phosphorylated, it gets dissociated. Now, E2F is free. So E2F is acting as a transcription factor, goes inside the nucleus, right? And results in the gene expressions of the cyclins A and E. Now, A आपका G2M में लगता है, E आपका S phase में लगता है, right? And it is also activating or activating in the PCNA, so that the replication is now speed up. Okay. The amplification or overexpression of D and 4,6 or loss of this complex or the inhibitor INC4, it causes increased RB phosphorylation and inactivation of RB function, thus leads to tumors. Okay. Simple. RB is the tumor suppressor gene. उसका अगर inactivation होगा तो obviously tumor suppress नहीं होगा. It is going to get active. Same thing, the same story here. Retinoblastoma is it gets phosphorylated, right? So when it's phosphorylated, E2F is free. It goes for the transcription. Okay. Now coming to the second checkpoint, that is G2 to M checkpoint. Okay. It is going to prevent the mitotic entry in response to DNA damage. Obviously, DNA damage increases or induces ATM-ATR pathway, activating check 1 and check 2. ये बहुत बार repeat हो गया. So by this time, you guys should be able to remember. Okay. So we have got the DNA damage now. DNA damage activating your ATR and ATM. Again, repeating, ATR is for the single stranded, ATM is for the double stranded. This is activating your check 1 and check 2 kinases. Now, this is going to phosphorylate your CDC25. Okay. It's a phosphatase. So what will happen? It will inhibit its activity and promotes its binding to 14-3-3 proteins to sequester it in the cytoplasm. This much is clear. P53 also activates P21. And both P21 14-3-3 proteins inhibits your CDK1 cyclin B complex. What is this? This is your MPF. Okay, by phosphorylation and sequestering CDC2 in the cytoplasm. Now, when P53 is involved in the dissociation of this complex by induction of GAD45, then it gets activated. Okay, time is getting over. Guys, this is clear. So P53 ने आपका P21 activate किया है. It is inhibiting CDK1 and cyclin B. इसका मतलब क्या? This is present in your G2 to M transition, MPF. Right? So it's all getting sequestered in the cytoplasm. Then the spindle assembly checkpoint and the progression to anaphase. Now, in the spindle assembly checkpoint, it is going to monitor the chromosomes on the metaphase spindle. Okay, so guys, this is your third checkpoint. So we talked about G1 to S, G2 to M. Now inside the M phase, this is the spindle assembly checkpoint. So as we know, chromosomes attachment is happening or not. Okay. Now once this is done, then only the cell enters from metaphase to the anaphase. Okay, to complete the mitosis. So the progression from metaphase to anaphase, it results from the ubiquitin mediated proteolysis of the certain key regulatory proteins, which is triggered by the activation of APC-C ubiquitin ligase. What is APC? So anaphase promoting complex cyclosome ubiquitin ligase. So it is inhibited by your CDK2 cyclin E and A complex during S and G2 phase. of the cell cycle. Obviously, right, that time I don't want a anaphase promoting complex मुझे नहीं चाहिए during my S phase and G2 phase. It should be activated in the M phase, right? So at the beginning of the mitosis, APCC is getting activated as a result of phosphorylation of your CDK1 cyclin B, MPF. Okay. So it remains inhibited, however, until the cell passes the spindle assembly checkpoint, after which activation of the ubiquitin degradation system brings about the transition from metaphase to anaphase and progressing through the rest of the mitosis. So clear, guys, what this slide is saying? That spindle assembly checkpoint is important because it will ensure that all the chromosomes are attached to the spindle fibers, right? Now, once this is happening, then only the APCC ubiquitin ligase gets activated. Otherwise, it is inhibited throughout your S and G2 phase. Okay. Now, this gets activated by the phosphorylation of your CDK1 and cyclin B. The spindle assembly checkpoint is remarkable in that the presence of even a single unaligned chromosome is sufficient to prevent the activation of APC. It means that all the chromosomes needs to be attached to the spindle fibers. Agar ek bhi miss ho gaya, APC will not get activated. Okay, so the checkpoint is mediated by a complex of proteins called mitotic checkpoint complex or MCC, which is formed at the unattached kinetochores and inhibits the activation of your APCC. Okay, now what is mitotic checkpoint complex consist of? It have got 4 proteins: BUB-R1, BUB3, MAT2 and CDC20. Okay, so they are present when the kinetochores are not attached to the spindle fibers. Okay, so 4 proteins are there: BUB-R1, CDC20, MAT2, BUB3. What are these? These are your MCC. Okay, now when it is not attached, then MCC is it is making or it is ensuring that APC remains inactive. Okay. Now the protein CDC20, it is required activator of the APCC, but its activity is blocked when bound to MAD and BUB proteins in the MCC. So the formation of MCC results in the inhibition of APCC. Clear? So CDC20 is required for the activation of APC, but CDC is bound with other MCC proteins. So जब तक वो उसके साथ bound रहेगा in the mitotic checkpoint complex, it cannot activate your APC. Once microtubules have attached to the kinetochores, okay, now the complex are no longer formed and CDC20 is able to activate rather than inhibit APC. So abhi kya ho gaya? So first it was like your chromosomes were present and there was no spindle fiber. So the MCC proteins like BUB-R1, BUB3, MAT2 and CDC20, they were present here. Okay, CDC20. Now once the kinetochore comes and binds to the chromosomes, so this complex formation will not happen. Okay, this results in free of CDC20. Now CDC20 is free, so it goes and activates your APC. Okay, now a activation of APC, it results in the ubiquitination and degradation of the two target proteins and trigger the metaphase to anaphase transition. What is happening? Ubiquitination, CDK1 cyclin B is there. Okay, so what will happen then? It is going to make it inactive by degrading your cyclin B here. Plus, it is also results in the degradation of your sectorane, making a separase active. Separase kya karta hai? It is going to make sure that the cohesin which keeps your cystochromatis together, जो यहाँ पे proteins रहते हैं, so that will be removed with the help of the separase, and that's how when it is ready to go for the transition from metaphase to anaphase. This much is clear. Should I repeat? Okay, I'll explain once again. So there are MCC proteins, okay, made up of 4 proteins, BUB-R1, BUB3, MAD2 protein, and your CDC20. Okay. Now, this MCC is present when the chromosomes are not attached to the spindle fibers. Okay. It keeps your APC inactive then. Now, once kinetochore comes and binds, this MCC complex is not formed. Okay. Now, when it is not formed, CDC20 is free, correct? This will go and now activate your APC-C. What is APC-C? It is your ubiquitin ligase. So it is going to ensure that the cyclin 3 is getting degraded. So the CDK1 activity becomes inactive. ठीक है? इतना clear है? Huh? So it means your MPF is now inactive. Also, it phosphorylates your securin. Okay, what will happen? This is now targeted for the degradation. Securin is bound to the separase. जैसे ये degrade हो जाएगा, separase आपका free हो जाएगा. So that makes it active. Separase क्या करेगा? It will go and degrade the cohesin. Cohesin क्या करता था? It used to keep at the centromere region both the sister chromatids together. Right? If you remember, we talked about the condensins and the cohesins. So पहले तो cohesins यहाँ पे भी रहते हैं. So that time they remove all this thing. Only at the centromeric region it was present. Okay. So now the separase is going to degrade cohesin. So it is now free. So एक spindle fiber यहाँ होगा, एक spindle fiber यहाँ होगा, and now your sister chromatids are free to be taken to the opposite poles. Clear now, guys?
Yes, ma'am.
Okay. Now, cohesin degradation is not catalyzed directly by APC. Instead, it degrades a protein called securin, and that is it is the inhibitory subunit of the protease called separase. So, जैसे ही आपका securin degrade होगा, separase activate होगा, which in turn will degrade cohesin. And cleavage to cohesin breaks the linkage between the sister chromatids. Here, what we have seen. Okay. Now, when all kinetochores are attached, tension is present. The MAD2 and BUB1 are free. Same thing hai, it's just that I got the different diagram. So as you can see here, APC is getting activated, CDC20 इसको activate किया है. APC क्या करता है? Securin is separase है, right? So here securin में वो ubiquitin tag add कर देगा, because of which securin will go for the degradation, and now separin is free. Separin या separase जो भी है, ठीक है? So it will go and जो भी cohesin present है between the sister chromatids, it is going to degrade those. So that is when this transition is happening from metaphase to anaphase. Clear? Okay, this is the same thing. So the separation of chromosomes during anaphase then proceeds as a result of the action of several types of motor proteins associated with the spindle microtubule. So this we have talked about, okay, motor proteins, dynein and kinesin. So once the cell has progressed to anaphase, APC also triggers the degradation of aurora and polo-like kinase, which will allow the cell to exit the mitosis and enter the interphase. So many of the cellular changes involved in these transitions simply the reversal of the events which is induced by CDK1, aurora and polo-like kinases during mitosis. So if you remember, CDK1 aurora को activate करता है, aurora polo-like को activate करता है, polo-like kinase activates CDK1. So this was the example of positive feedback. Now, for example, when it is going, like reversing the activity, so it means what? आपका nuclear envelope reappear होगा, chromatin condensation की जगह decondensation होगा, right? And the microtubules which were present in the spindle fibers which was formed, okay, that will be depolarized and normal microtubules will be formed, okay, as a result of these loss of the activity of the mitotic kinases and the dephosphorylation of the proteins. So if you remember, जब आपका MPF activate हुआ था, there were 4 things happening, right? Nuclear envelope degradation, Golgi apparatus proteins degradation, then spindle fiber formation, lamins ka phosphorylation hua tha, right? So all these activities now going to get revert, so that the cell is exiting the mitosis and entering the G1 phase again. So both the daughter cell will enter the G1 phase then. Now, what is anaphase promoting complex or the cyclosome? So it's a multi-subunit E3 ligase. It induces proteolysis of different cell cycle regulators. So here you can see so many complexes are involved. E1 is there, E2 is there, and E3 is there. Okay. No need to remember. So that's okay. कितने हैं, कितने complex, कितने subunits है, that and all you don't have to remember. Just remember what is the function of the APC. So just to summarize, it induces a transition from metaphase to anaphase by tagging specific proteins for proteolysis like securin हो गया, M and S cyclins हो गया. Okay. Now during metaphase, cohesion mediates the binding of the sister chromatids together. Now when this gets activated, आपका securin जो है ubiquitin degradation के लिए जाएगा, separase आपका active हो जाएगा, ठीक है? So that then it is going to degrade the cohesin. Same thing. I'm just skipping now. Now coming to the cytokinesis. The completion of mitosis is usually accompanied by cytokinesis, giving rise to two daughter cells. Okay. Now cytokinesis initiates shortly after the anaphase is triggered. In contrast, aurora and polo-like kinases remain active in the anaphase and play important roles in coordinating nuclear and cytoplasmic division of the cell. Okay, so cytokinesis of yeast and animal cell is mediated by the contractile ring of actin and myosin filaments, which is formed beneath the plasma membrane. So this you know. So with the help of the aurora and the polo-like kinases, the contractile ring is formed. Okay, and the cell is eventually cleaved in a plane which passes through the metaphase plate perpendicular to the spindle. So this and all, yeah, so it will ensure that both the daughter cells are getting half up. Okay. Now in the case of the plant cell, thoda sa different hai. If you know about it, there is a cell plate, right? Instead of a contractile ring, cell plate is formed. So the mechanism of cytokinesis is different for higher plant cells. Rather than being pinched in half by contractile ring, these cells divide by forming a new cell walls inside the cell. Okay. So in early telophase, vesicles carrying the cell wall precursor from the Golgi, they associate with the remnants of the spindle microtubules and accumulate at the former site of the metaphase plates. So what is happening? The Golgi is coming. Okay. So they start coming to this area where your spindle fibers are present, and then they start helping in formation of this cell plate formation. See, as you can see, this is your cell membrane, cell wall is there, right? So this Golgi will start helping in extension of this particular thing. So ये ऐसे बनाना शुरू कर देंगे. Okay. So these vesicles then fuse to form a large membrane enclosed disc-like structure. Their polysaccharide contains assembled to form the matrix of a new cell wall, यानी कि cell plate. So slowly they start forming like this. ये यहाँ पे fuse हो गया. The cell plate expands outward perpendicular to the spindle until it reaches the plasma membrane. The membrane surrounding cell plate then fuses with the parental membrane, and totally what will happen at the end? It is going to fuse it like this. Okay? Now, connections between the daughter cells are formed as a result of incomplete vesicle fusion during the cytokinesis, and that results in the formation of your plasmodesmata, which also we have discussed earlier. Clear, guys? In plant cell, how it is happening? So it is not that यहाँ पे contractile ring बनेगा. Okay. So instead of that, Golgi vesicles comes together. They start, taking off these portions like this. They start fusing like this. Cell plate formation, and then slowly they fuse. Since there is no complete fusion, because of this, the plasmodesmata is formed, which results in the connections between the two cells. Now, what was the analogous to plasmodesmata in animal cell? Akshaya? Analogous to plasmodesmata in the animal cell? Tanvi? Which results in direct connections between the cytoplasm of the two cells, of the two adjacent cell.
Connection?
Yes. So those are the names of the proteins, but the real answer is, the correct answer is gap junctions. Okay. Gap junctions are made up of connections. Okay. That's all, guys. आपका cell cycle यहाँ खत्म होता है.